operator. Operator, you can play it whenever you're ready. Okay, thank you, operator. Okay, ladies and gentlemen, before we start the main event of the day, let's open it with the pray according to each other's beliefs so that every insights that we got from today will be beneficial for us. Okay, thank you. The next agenda will be the report of today's event that will be delivered by Mr. Agung Sedayu. Mr. Agung, can you hear my voice clearly?
Yes, thank you very much, Tatu.
Okay, please. Mr. Agung, the time is all yours.
Ya, Assalamualaikum warahmatullahi wabarakatuh. Ibu Dr. Reni Indrayanti, the
Waalaikumsalam warahmatullahi
coordinator of biology program at the UNJ, the teaching staff of the biology program, our speaker, Dr. Ewan Low, and students. Allow me to report to you all about this talk. This talk is called the Teaching Practitioner. That's the name, where in this event, we invite a professional, a guest speaker, who is a professional in a certain area. And in this semester, we invite, in accordance to our course, the biodiversity and the systematic of the phanerogams. So here we have Dr. Yi-Wen Low from Singapore Botany Garden. This talk is attended by about 80 students who are enrolled in the course. And also we have some 18 students who are not in this course, enrolled in this course. We have also several academic and teaching staff attending this course. I wish to thank many people who participate in this talk, our program coordinators, our speaker, our teaching staff, our students, and especially we have a small group of student organizer, 5 people that are so keen in organizing this talk. I thank you very much for your contribution. So I think that's my report. So without further delay, I give this Zoom back to Tatu Mutiara Shiva. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much, Mr. Agung, for the report and also the greeting. Now we will hear the greeting for Mrs. Reni Indrayanti as head of biology department. Ibu Reni, can you hear my voice clearly?
Yes, I'm here.
Ibu Reni, the time is yours, please.
Okay, thank you, Tatu, as a moderator. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning to our student, lecturer, and our special guest, Dr. Yiwan Lau. It is a pleasure to welcome everyone to our fifth one meeting lecture. Hopefully, today we are in good condition. We want to thank to all the participants that are joining to this lecture today. As Pak Agung said, that this is we call a practitioner lecturer. We also thank to Pak Agung and all the team that who's coordinated this session, and a special thank to Dr. Yuan Liu Lo or Dr. Lo from Singapore Herbarium Botanical Garden as our guest lecturer today. According to the discussion and the team, Dr. Law will be presenting exciting topic about taxonomy on diversity of the close genus Syncytium myrtaceae and the kaca piring genus Gardenia rubiaceae in Malaysia. I think this topic is very interesting, and hopefully that it will enlighten our knowledge, our students' knowledge about botany in our course, especially in biodiversity and systematic of panerogamy. And I hope that this lecture will also inspiring our students to conduct research on biodiversity of plant, especially for endangered species in Indonesia. And hopefully Dr. Riel will be our, maybe have a time to help our student if they have a research about the botanies. I think without any further, we would like to give to thank you again to our student and our lecturer for attending in this session, and we would like to give the time back to the moderator. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much, Mrs. Reni, for the warm greeting. And now we will have a presenter introduction. They will be delivered by the one and only Mr. Agung Sedayu. Please, Mr.
Thank you, Tatu. You're so kind. So allow me to chair this session. And before we start the session, we'll have 2 points to announce. First of all, it's about the regulation of our meeting. First, we have about 1 hour presentation and 30 minutes of question and answer session. During the first session, I suggest that we keep our microphone off, so we're not interrupting the presentation. And during the 30 minutes question and answer, I think it is possible and it is okay if you turn on the microphone and address your question directly to Dr. Low, or alternatively, you can also address your question via chat in this Zoom room. Secondly, I have to introduce you to about our speaker, Dr. Yiwen Lo, before he starts his presentation. Dr. Yiwen Lo is a tropical botanist at the Singapore Botanic Garden. Whose interest is about taxa which are very dear to us. He is a keen observer and researcher about the coffee Rubiaceae family, also Bignoniaceae, and Jambu, the Myrtaceae family. I count from IPNI website that Dr. Low has already described and named about 20 new species. One Adonidia, Arisaema, Begonia, Melastoma, Macrosolan lasianthus prismatomeris, 3 species of Syzygium, Jambu, and 6 species of Gardenia, Kacapiring. So definitely today we have an honor to have a renowned taxonomist who understand the family of plant in our area, Dr. Yi Wenlo, to talk to us. In order to have many species which bear his name behind the species name, 20 species. Dr. Low has extensive collaboration with lots of scientists from internationally or regional area. He has collaboration with Brazilian, with Indonesian. He has collaboration with Brunei scientist, Dr. Joffrey. I know him quite well. Well, I met Dr. Joffrey last time, 2013, I guess. And he made I think Dr. Low had made extensive what do you call that, collaboration with all those people. So I think the student can make him as an example about how to do research with collaboration, which is important in this time of era, I think. So if you are interested in Dr. Evenlow, I think you can search in the National Park of Singapore website, and also you can check the Google Scholar about his contribution to the world of botany internationally. And without further delay, I present you Dr. Yi Wen Lo. Dr. Low, the Zoom is yours.
Thank you very much, Agong. Hi, very good morning. Can you hear me loud and clear?
Yes, it is very clear.
All right, thank you so much. First of all, a very good morning to everybody at the university. Thank you so much, Agong, for the invitation, the MC, and everybody who made this lecture possible. I'm really very honored to be invited as a speaker to share my very short experience. I wouldn't say it's very rich, but I think it is very exciting for the past 20 years, I must say, since I did my degree. So without further ado, I will just start my talk. So this is a very brief background. I'm a Malaysian, so I speak Malay, but I think my Bahasa Malaysia would be very different from Bahasa Indonesia, and I am embarrassed to have an official presentation made in Bahasa, because I don't think my bahasa is very polished, as most of the Indonesian would do. So I have developed an interest in plants since very young. I believe the students who picked up biology courses in Universitas Negeri Jakarta would be the same as well. I have always wanted to be a botanist when I was very young, but I couldn't get into a university degree that offers botany, because by the time I signed up for a degree, the climate was very different. The universities are no longer offering botany courses as a degree being awarded, but there were a lot of others, biological aspects courses being offered. So I was given a forestry program to attend to in Malaysia, which I took it, and I really enjoyed my time in Sabah. That's where I developed even more interest in orchids. I've learned so much. After my forestry degree, I went back to University Malaya to KL, and I signed up for a master's. And I was also against the odd, in a sense that for most students of my time, they would sign up for a botany in a sense of coursework where you will finish in 1 year and then you move on to maybe a PhD or you look for a job. But I took the odds. I went for a bio-research coursework in botany, and I was also working part-time as a research assistant in a university, assisting my professor then. And I kind of finished it about 5 years doing part-time for my MSc. And that's when I did a lot of my work on gardenia. And after my MSc, I pursued I took a job in Singapore, and I started off as an arborist only for a few months before I was officially transferred to the herbarium and become a researcher, which I'm being trained for. It was only much later, around 2017, that I was offered a scholarship to pursue my PhD. And I'm very thankful to National Park Sport Singapore for the opportunity and the investment they invested in me in botanical sciences. So as Agong has shared with you, this lecture would be divided into two parts. Two main parts is actually revolve around my work. The first part, it will be on the genus Gardenia, or the Cachapiring genus of the coffee family. And the last part, I'm afraid it's going to be a bit heavy, but I will try not to emphasize too much on the technical or the technicality of the work, but share in a layman term of what I have done for my PhD studies. And then a bit of recap and question and answers. Okay, so here, just to share with everyone, I'm here in Singapore, in this very tiny island, and you guys are here in the Java island, and this is Southeast Asia. You have to, I'm sure all of us will know this, but just remember this scene. And of course, we have all the countries now. But then in this very exciting area we are in is what we call Malaysia. And this term is actually coined by Zollinger, where he kind of recognized the area in gray would be what he calls as Malaysia, where we share a lot of similar flora and fauna especially. Okay, but of course, you would also know and heard about the Wallace's line, where it cuts through in between Borneo and Sulawesi, where Wallace observed that there was this transition of animals and insects that he's seeing and he's experiencing in the field. Okay, but in terms of plants, we have Van Steenis, who actually made these three very important notes. The first note is between Malay Peninsula and Peninsular Thailand. If you can see my cursor here, and then the second node is between mainland Asia, Taiwan above on the north, and Philippines. All right. And the third node is between Australia and New Guinea at the very tip, Cape York. All right. So just before this talk, I was away on a 3-week field trip recently in Peninsular Thailand. I have experienced myself of a particular plant genus called Brachytomi in Rubiaceae that I have collected it long time ago from Cameron Highlands in central Peninsular Malaysia here. But then when I collected it here in Botany, and I realized that this is again a northern Asiatic element that actually intrudes or travels down from this narrow neck and arrive at the central highlands of Peninsular Malaysia, but it doesn't go down to Singapore, for example. Brachytomi doesn't occur down south, further south from Cameron Highlands. It is really important to be able to see this for ourselves in the field, as well as looking at it from papers and specimens. So here, just to highlight what is Malesia, according to Johns in this very early work presented in Flora Malesiana, he estimated around 45,000 species of only vascular plants or flowering plants occurring in this region. And of course, this region, as you have seen earlier in my figure, it goes all the way from Peninsular Malaysia to New Guinea. And we all know today it is divided into, previously it was 6 countries. Of course, now we have Timor-Leste, so it's 7 countries. But I want to stress here, this is a very important note, is that the bulk of the Malesian region, this very rich region, is in Indonesia. It's not in Singapore, it is not in Peninsular Malaysia or in Malaysia, it is not the Philippines, it is not Brunei, but it is Indonesia. All right, so this is the first topic that I would like to dive into. This is, as what my PhD supervisor said, this is my first love, because this is the first plant that I was very much involved with. So this is gardenia of the coffee family. Here I'm showing you a varieties of flowers and fruits, mostly from the Malesian region, except for a few. Okay, but it is all about resources. When we talk about plants and the importance of plants, we try to highlight the resources and uses. So for gardenia, we know in English, the common name is also called the Cape jasmine. it's because of its fragrant flowers. And then humans started tapping into this natural resources, and then develop into all these perfumes. And this industry itself is worth up to about $10 billion, this perfume industry. But of course, the other side of beauty, the contribution towards the beauty products and luxury products is health products. The Chinese, and I'm sure also other civilizations, have documented the importance of gardenia fruits. And this dry, what I'm showing here is the dried fruits of Gardenia jasminoides. It is used in Chinese sensei shop, where the fruit is boiled, and if you soak it in water, you get it this very rich red orange dye. Okay, this dye, this color is actually comes from the compound called crocin. Okay, which is a crocin carotenoid constitutes. Crocin is very popularly known in saffron. I think many of us would know saffron. Of course, we use it in our delicious nasi biryani, and I'm sure in Japanese cuisine or Indonesian cuisine, where there could be a different usage of crocin. So the crocin comes from the crocus flower. They are actually the dried stamens. And if I'm not wrong, there is a particular term to these stamens. They call it something like the goal of a particular flower, because saffron is very expensive. If you go to a supermarket and you look for saffron stamens, dried saffron stamens for cooking, it costs quite costly. And coincidentally, crocin is also known to occur in jasminoides fruits. Okay, and it has been recorded with all these therapeutic uses. And I have always been very careful is that when we share something about users and for human consumption, I'll always try and provide the references, because this is what I am quoting from, and it's not a hearsay or an old lady's tale that I'm sharing. Okay, so gardenias in Malaysia that I have experienced are mostly shrubs and trees, which I will show in the next two slides. But when I got the opportunity to travel to Australia and work with my friend here, Bruce Gray, a retired staff of the Atherton Herbarium, very, very knowledgeable with Australian and Queensland plants. I was when I went over to Australia, I said, "Oh, I'm interested in gardenia. Would you be able to show me around?" And he just took me to this place in Mossman and showed me where they are cultivating this gardenia as street plants, but naturally it occurs in the bush, they call it, as a prostrate herb or shrubby, lazy plants, where it doesn't grow erect into a tree or bush. But and I was really amazed by it. So this is to illustrate the habit. Okay, but this is what our typical gardenia from Malaysia would look like, that you would know. So this is Gardenia mutabilis, and the other one being Gardenia jasminoides, the kacapiring itself. This species has a very interesting story behind it that I must share with all my fellow Indonesians. What we have here, the local name of this gardenia is known as Thai gardenia. I think the error comes from sorry. I think the error comes from the book that was produced by Singapore, which is the 1001 book, where a horticulturist may have obtained a clone of this plant from Thailand, and not knowing or not having a common name for the horticulturist is a big problem. So they decided to give it a name called Thai gardenia, which was very, very misleading. Gardenia mutabilis is native to Sulawesi. It also occurs in some islands in the Philippines. But I am very certain and very sure that this plant could have been obtained by a nurseryman from Thailand in Java, brought it back and propagated it well in Thailand, and then somehow marketed it to Southeast Asia, marketed it in Southeast Asia. So when I was revising this genus, and I was very puzzled with the name Thai gardenia, that I couldn't get it off my head, and I've been trying to correct this over and over again, even in Singapore, even today, that we shouldn't call this a Thai gardenia, or we shouldn't even give this a common name. We should just simply call it Gardenia mutabilis, which is a beautiful plant, is a shrub. And then, of course, gardenia can be a big tree, very tall trees in the rainforest. What we have here is Aslan. This is a photograph taken from Brunei in the Balaik forest, a pristine forest. And for the very first time, I have seen this species many times, but sapling, because they are very easy to recognize in the field when it's sapling. But I've never seen a tree so big. So of course we can't obtain a material so high up on a tree. I did, however, took a photograph of the crown where you can see the fruits. And of course, how I chanced upon it is that because we found fruits that were half eaten by maybe primates, and they were all discarded on the forest floor here. And when we pick it up, and I was like, hey, this is Gardenia bumenia. And true enough, we saw the tree, we found the tree, we traced it to this grand tree here. And I have never seen gardenia with a fluted, somewhat very low fluted buttress like this. Maybe because I have really never seen a very, very mature gardenia. Okay, so brief introduction of gardenia. It was named by John Ellis after a naturalist based in Charleston, Alexander Garden, and it is typified by Gardenia jasminoides. Okay, and the material was said to be collected from the Cape of Good Hope. All right, it was named in the 1700s, and the material was from the Cape of Good Hope. Again, this is a very clear story of plants being cultivated from very, very early on. What we know today, Gardenia jasminoides, is restricted to southern China, southern Japan, and maybe northern Vietnam region. It doesn't occur naturally in Africa. But of course, you know, in those days with all the ships traveling and people carrying plants around, like the chili, like the potatoes, like the sukun itself, a lot of us have actually altered, humans have actually altered where plants do occur naturally, and this is a perfect example. Another example, another evidence of it being a cultivated plant is that the type flower is a double flowered form. So if you look at the flower, it has multi-petals. Typically, gardenia has only a single petal. So again, this type material itself is from a cultivated clone itself. All right, so Gardenia is quite a big genus with up to about 200 species, and it only occurs in the old world, from Africa all the way to the Pacific Islands in Hawaii. All right, in Malaysia, we have about 40 species. Okay, a bit of a taxonomic history. Gardenia is like a dustbin in the past, where if you have a certain characters, it will be grouped in that genus. So up until very recently, before Key started looking at it, Gardenia is extremely heterogeneous. You can have Gardenia with spines. Then you have Gardenia with no spines. Then you have Gardenia with single solitary terminal flowers. And you also have Gardenia with an inflorescence, flowers with 3 or 4 or 5, 6 flowers in an inflorescence. All right, so Ronald Key was the first one who kind of study what is a gardenia for Africa, and his breakthrough actually served as a catalyst for Tavan Gadam, a Sri Lankan botanist, to work on South and Southeast Asian gardenia, and then followed by Kun Ming Wong, who did it for Malay Peninsula. But then that was back in the 80s, and we really need to revise the genus. But then again, don't forget, the genus itself is 40 species in Malaysia. So it's very big for anyone to want to make it a research subject. So in order to frame the kind of framework for my study, we try to scale down so that we will not have too many species for a young student, me, then to pursue, because you will then never finish your study. We want a student to graduate and to move on. So that was the aim of my supervisor. So therefore, we frame it in such that we have a good framework in which we are only looking at the Sundaland gardenia. We're interested in the taxonomy. Most of my work would be based on herbarium specimens with a little bit of field work. But over the years, ever since I joined Singapore, I was able to travel and I was able to see more. But before that, I think my fieldwork was very restrictive. It was only in Peninsular Malaysia. I did some fieldwork in Sabah and Sarawak, and also went one trip to Brunei, and that's about it. But I was very fortunate that I was able to make herbarium specimens on loan from Kew, from Bogor Herbarium in Cibinong, from Leiden, from Bangkok Herbarium, and from Edinburgh as well. All right. So besides that, I also did a little bit of molecular work. I will not touch more about it because it was really, really rough. I will tell you more later as we go on. So why Sundaland? So Sundaland itself is interesting in a way that if you look at this figure here, the dark outline would reflect our current situation, where we have the Borneo Island and Peninsula Malaysia, Sumatra, Java, and etc. But the lighter gray area reflects what would be a connected land mass in the past, million years ago, during the last glacial maximum, when the temperature is much lower. So when the temperature is much lower, water that we are seeing today, the sea level that we are seeing today is much lower, because the excess of what we are seeing today has been locked up as ice in the North and South Pole and many areas. That's why we have low sea level and a much greater land mass. So can you imagine this piece of this area was connected in the past? And of course, plants can move around freely with no barriers, no sea to cross, and that is why it is important when you start a study that you are not restrictive to a particular area, but you also be mindful and think outside of your region. Not too much, but enough and reasonable to have a good biological understanding of your study subject. All right. So this is a summary of what I have done. I have roughly examined about 1,500 sheets, herbarium sheets, from various herbaria, 18 altogether, and I can define gardenia as having extra axillary branching, which means the branches doesn't occur from the nodes, but the branches occur slightly above the node. So if you find a kacha today or after this talk, you can have a close examination to see the branching. Where does it start? I can assure you that it starts above the nodes and not at the nodes. Okay, and then stipules is interpetular. When it's dry, it's connate it's a connate into a tube, but when it's dry, the stipule remains, and therefore it stays on the twigs, and it becomes loose, and you will always see that it is almost like a ring on the twigs of the tree. Resin is always conspicuous. It's present at shoot tips. You will always see it, but of course there are exceptions, which I must say, Jasminoides and Mutabilis would not have a very clear, conspicuous resin, but you can tell from the leaf layer that it is glossy and shiny, and it is coated with resin. Flowers is always solitary and terminal. It will never be an inflorescence. Corolla is hypocrateriform, white, darkening to yellow, and then eventually to very deep orange. Again, there are exceptions. I will show you later that kacha piring is a very good example of Gardenia jasminoides. It doesn't change to yellow as you would see. You would know what I'm talking about. And then, of course, secondary pollen presentation. All right, so here, this is what I would say exudate the resin. It is typical to see resin in gardenia like this. It's either clear or it can be very thick and opaque, like these two examples here, yellow, but very, very rarely it is, very, very rarely it is black. So this is a very good example. This is Gardenia bemania with the black resin, which I have never seen for any other gardenia apart from this particular species. And this is what I would call an autapomorphic character, which only reflects to one species. Most would have this opaque yellow, clear, but in the sense of jasminoides, you won't be able to see it, but you can tell from the very glossy surface of the stipule and the leaves. All right, so here we have jasminoides. So I have grouped them in the sense that the two images from the top, the two images from the top are actually species that doesn't change color much after anthesis or post anthesis. So jasminoides and scabrella on your right, jasminoides on your left, they have white flowers at anthesis and it remains white. It may change to a slightly creamy off-white, but otherwise That's it. It will never turn yellow. But the species at the bottom, so I have from left to right, Mutabilis, Graffitii, and Carinata. When they bloom at night, it only blooms at night. It will be white, just like Jasminoides. But by the next day, daylight, it will be pale. It would have turned to pale yellow. And then by noon of the same day, it would be yellow. And then it stays yellow for maybe another 2 more days. And it eventually becomes very deep yellow-orange. All right, so here is different stages that I've documented of Carinata. And then this is what I would say describe as a secondary pollen presentation. So what we have here is the flower of Gardenia jasminoides. The flower bud is on your left, and on the right is artificially forced open a flower bud at the very early stage of just about anthysis. Okay, I cut it open, and what we can see, what we can observe is that the pollens have already been deposited on this non-receptive stigmatic surface of the flower. So when the flower is fully bloomed, the stamens are not, I mean, the pollens are not presented at the stamens, but rather it is presented on the stigmatic surface. It's non-receptive. So insects will come, bees will come, and they will get dusted around, when they try to obtain maybe nectar at the very base of the corolla tube. And then all this will be kind of like rub off, the pollen will be dusted off on the body of the insects. And then eventually, of course, then you have other elements like rain and wind. So when all that impacts on the stigmatic surface, it comes to a point that the stigmatic head will become receptive. And then when it's receptive, by the time it is receptive, all the pollen has already been taken away or gone. And this is when it is waiting for an insect, perhaps with pollens from another tree being dusted on, to be introduced to this very receptive stigmatic head. You can tell that when it's receptive, they're actually swollen, and then the grooves in between the stigmatic heads are actually quite far apart. Okay, so some findings here to share. Stipule morphology is a good character for the Malesian species. For the Sundaland species, we can tell that there are two forms, which is cup shape and a sheathing tube. So here I'm showing you on the left, the top and bottoms are the cup-shaped stipule. So here is the stipule. Of course, these line drawings were prepared from dried herbarium specimens. So what you are seeing here is that the twigs have already shrunken. In real life, I've seen it in a while. The stipules are actually sheathing the twigs exactly tightly, and that's about it. It will never shrink so much as as it is drawn here. This is actually drying artifacts of the materials. And then if you can see the cup-shaped stipule from the top figure, the mouth is actually truncate. It hasn't got any lobes. But if you look at the stipule at the bottom, so this is Gardenia Graffitii, you can actually see 4 lobes. They are actually lobed around the stipule. And this would be a kind of a good character. You won't be able to tell the species based on the stipule, but you can group them to a working group that you can sort out later. Once you have a working group, then you can kind of match the leaves and all that if you don't have fruits and flowers. All right, on the right, we are seeing is what I would say Gardenia thailandica, where you have a sheathing stipule. So I kind of given you an image of it where the mouth is actually oblique, and as the stipule pushed extends and matures itself, it pushed open, and it forms more like a kind of like a slanted mouth or calyx tube. I mean a stipule surface. Then calyx morphology is very, very important in species distinction. So I receive images all the time from colleagues asking me, oh, can you help me to ID this gardenia? And most of the time, the image that I receive is a close-up of the very beautiful flower, the lobes, and the corolla lobes, and the stigmatic head, which obscure everything, the most important part, which is the calyx part. And I always have to tell them, you need to show me the calyx because it is very important to be able to tell the species. I need to see the calyx tube, not the beautiful flower. And here is an example of all the Sundaland gardenia that I have commissioned the artists to prepare of the different types of calyx ornamentation characteristics of the calyx tube. So you would see some of the tube is quite long and narrow. Some is short and flat open like a trumpet, while others is again much shorter, some with wings. This is what I would call triangular wings, whereas this particular species here, subcarinata, would have keels. So it has got ribs that extends to keels above the calyx mouth margin. And then, of course, you have Chania with ridges on the right side, the farmost right side, Gardenia Chanii, and then of course below it is Tyrocalyx. So you can tell that this is the most important feature apart from the rest. And of course, it was a fashion of the lab of the professor that I'm attached with that we were also very keen in looking at tree growth architecture, meaning to study this, you need to look at intermediate trees to very young growing sapling, but you can't tell from a tree in the forest of very big trees. You can't tell that. And to learn more, if you are very interested, is that you should look for this book by Hale, Alderman, and Tomlinson, 1978. It is a very old book. It is also a very good book that I have even purchased a PDF copy. So, and it's the entire book. And I'm happy to share if people are keen. You can write to me and I can send you a PDF of the book. Of course, I have a printed copy of itself for myself, but I'm really happy to share because this book is out of print. It's very old. You can of course buy it online, but otherwise, if you need it and you would like to learn more, you can write to me and I can send it to you. It's no problem. It's not bad for me. It's bad for the publisher, but it doesn't matter. I think Springer earned a lot of money from it already. So I have looked at about 12 species to study tree architecture, and we recognize two models. The first model is Leeuwenberg's model. You can find out more from the book. What Leeuwenberg's model is that it is typically modular, autotropic branching for all parts, meaning the leaves are facing upward the sun all the time, against a certain portion of the leaves where it is parallel on a branch, which will be on a different models. So this would be Leeuwenberg's model, and it is restricted to the bush, to the species that has got a bush habit. And I studied jasmine orchids from the pot, which you can see the branching itself. This is 3D, of course, and then the line drawing in the text is 2D. So it's I must admit it's very difficult to decipher, but if you have used it often and you try and understand a bit more, you can actually picked up the models. There are about 28 models recognized by the authors, and you can fit most of our tropical plants, I mean trees and even climbers, to their models, their proposed models. And then, of course, all our trees are in the Scarron's model, where you have an autotropic trunk, but a plagiotropic branches. So the branches, at the branches stage, you would see the leaves are not arranged in a sense of a certain angle facing the sun, but they are actually parallel facing the sun. So that's more that you need to read and follow from the book about autotropic and plagiotropic branching. So again, I provided a photo here to share. This is actually Giardinia thailandica from Langkawi that I have observed in a while. And of course, from my MSc work, we have a lot of publication. My revision, unfortunately, it's still not published after 11 years. I haven't got the time after my MSc, started working and there was a lot of commitments. Then I started my PhD, and the revision, unfortunately, was not published until now. I will try to kind of get this done within the next 2 years, where a bit more things will be polished. We are also trying to start a new work looking at the phylogenomics of gardenia with other collaborators, which I'll share more. So then, of course, I was required to, as part of my MSc, I was told that, it is then a fashion that you must also do a bit of molecular work. And I did it, looking at 3 regions, 2 chloroplasts and 1 nuclear. These are actually very, very out of date now today. If you want to do it as an exercise, I think it's great. But if you would like to pick this up and publish it in a current setting, it will be very difficult to be accepted, because people have kind of moved away from Sanger sequencing and into whole genome and looking at more genes rather than just 1 or 2 regions. We are aware. So I can show you the results. So for my molecular work, I have about 16 tips with 3 outgroups. And of course, this is, then I have the combined chloroplast analysis, and then I have a separated ITS, and then I have a final analysis in which I'll combine the chloroplast and the ITS, and then we kind of summarize the outcome. If you look at the trees now, they are all poorly resolved. There's nothing much you can tell, and it doesn't mean much. The story will change if we move further. So the next phase that I'm very keen to do is in collaboration with Professor Victor Eber, whom I have worked with for my PhD. to move towards phylogenomics of gardenia, where we are keen to look at about 100 taxa. We are first, of course, we need to find the funding, the money. It is the same problem for all of us, and also maybe for the staff at the university. That for any projects that we always need to first look for funding to support this. We are hoping to get some money from America. We are hoping to get some money. I'm hoping to get some money from Singapore, but we are not sure yet. But we are still moving forward. We started off with a pilot of 10 gardenia. We have sent it for sequencing. We have now obtained the sequences, and we hope to kind of assemble it over this summer in America, and by a student of Victor, and then maybe we will know a bit more stories. So I'm very keen to study about the Jasminoides and the other species, the shrub species that doesn't kind of change color. Even the fruit itself, they behave very differently for some of these species. All right. So, okay, to go back, Sundaland, here is a list of species based on my revision. We are recognizing 12 species. We have 2 new species. In fact, the species number 12 is undescribed from Mount Sangkulirang. Mount Sangkulirang, it's a limestone mountain in Kalimantan. There is a sheet that I have seen from Leiden and Bogor Herbarium, which is the most beautiful. It is a shrub. It is not a tree. It is a shrub. So therefore, it will goes into the Leeuwenhoek's model, and it has very small leaves. And this is definitely new, which I haven't described it yet. And the other one is a new variety of likely from Mount Kinabalu. And then worked with my former supervisor, Kun Meng Mong. We kind of look at the Philippines gardenia, and I received loan of materials from Bogo. And then when I examine and look at the calyx characters, I found new species from Sulawesi. That's when I published and described the 2 new species from there. And then based on the materials that I have at the moment, we have still new species from Halmahera, as well as Flores to describe. So it's about 3 there, but we don't have kind of like a big framework for this region. All right, so I don't know, maybe somebody from the university would be keen, a student would like to pick this up as a project. I'm happy to pass this on, maybe for somebody from the university can supervise a MSc project or undergraduate project. I have no idea, but I'm happy to talk further with anybody who would keen to develop more. And here, this is another interesting place. I've been to New Guinea only on the West Papuan side, and I visited 4 times now, but I don't think it is sufficient. So New Guinea is the largest tropical island in the world. The terrain is very difficult to get around, and it takes time to get from one destination to another. So I don't think what I have seen is sufficient to understand the flora there properly. It requires a lot more dedication. It requires a lot more time to do. And I think it is also important that the local Indonesians should also kind of pick up this region. This area is very, very important. It is pristine now at the moment. Still not much destruction, unlike Borneo or in Sumatra or in Peninsular Malaysia. New Guinea is still very much intact. and this would be a very important areas to look into for further research. So again, an opportunity here, to see if anybody will be keen to work on. Based on the herbarium sheets that I have examined, gardenia in New Guinea seems to occur for some species as an epiphyte, which our preliminary look at the species, we think that it is not a real gardenia. It could be, something different. It could be even a new genus. Again, we are hoping to include this in the molecular work that I'm working with Victor, and that will be very, very exciting. All right, second part of my talk. This one is going to be very, very heavy. I will try to simplify a lot of steps and then move on. And you can ask me during the Q&A of this topic whatsoever. All right, so just something to share. The top 3 genera of trees in the world actually belong to Myrtaceae, and Syzygium is the top, followed by Eugenia and Eucalyptus. All right, and then when you look at the largest tree genus in New Guinea and Borneo, which is the largest tropical island and the second largest tropical island, the tree genus is Syzygium. So I have participated in work by Rodrigo Camara-Leirit, which was published in 2020, where we kind of generated a checklist, and my supervisor at Q, Eve Lucas, and I, we were tasked to look at Myrtaceae. And we were very surprised. I mean, we are not surprised, but we were just overwhelmed by the figure. It was kind of over 270 species in New Guinea alone. And we are talking about known species, and there could be even more. And in the same report by Beach et al., where they talk about the tree diversity in the world, Indonesia is one of the top 3 countries in the world with the most diverse tree flora. The other being Brazil and Colombia, and they are in South America, which means Indonesia is the only Asian country with the most tree flora. This is how important Indonesia is. When we talk about Flora Malesiana, we cannot talk about Flora Malesiana without involving Indonesia. So to better document our flora, we should encourage our colleagues and our friends in Indonesia to pick this up, that you have this wealth in your country that we must protect, we must know more before we lose them all with all our developments. I'm not against development. We need development, but we need sustainable development, and we also need careful development for many parts of the country. So the genus is typified by Syzygium caryophyllatum, which is a Sri Lankan species, a tree species. And Syzygium itself is derived from the Greek word called syzygios, meaning a partner. So many of us who worked on Syzygium kind of like wondering, what are the meaning or the partner. So we kind of speculate that it could be referring to the paired leaves. All right. Based on POVO, there's about 1,200 species, but there are also other work that says it's close to about 1,800, but I'm not sure. Again, Syzygium is an oval genus. It occurs from Africa all the way to the Pacific Islands. But with this broad distribution, the center of its richness is in the Malesian region. And therefore, when we talk about Malesian region, the center of richness of Syzygium is in Indonesia. All right. Due to its immense diversity, it is exploded. It's like an explosion. Syzygium becomes a kind of like a taboo plant group that people tend not to look at. To be very honest, when I was working on gardenia and I was doing my fieldwork in Peninsula Malaysia and Sabah and Sarawak and Brunei, we tend to come across lots of Syzygium. We will collect, but we will never make an effort to try and ID the material because it is just so difficult. So it will always end up the mounted materials in the herbarium as Syzygium sp. So if you don't believe me, you can go to any herbarium. You can go to Bogor herbarium. You can go to the Syzygium collection and you look at the amount of unidentified materials. It is really, really a lot because nobody dares to work on it. And that's why many of us say Syzygium is very difficult. And it is difficult not because of, lack of characters is one thing, but it was also a very complicated history in the past where it is kind of mixed up with eugenia. So I have a friend in Peninsula Malaysia, a nursery friend. He's in his early 70s. He's a very good nurseryman, and he would still continue to tell me, I have this eugenia and that eugenia. Is because he has been taught that way, that or introduced that Syzygium is eugenia in the past, and it's not surprising. So I have an image here of a eugenia and a Syzygium placed side by side. In the past, they were all just grouped together. Everything is called eugenia. They all look so identical. But of course, there are other characters that they have looked at, especially the ovules. And even now, the molecular studies have shown that they are two completely different groups from different lineages. So this is a work that I'm also part of with Olivier from Q, kind of led by Yves Lucas as the senior author, looking at the entire order of Myrtales. What we know for sure is that in this study, as has been shown in much earlier Sanger sequencing work, so this is a next generation sequencing work, HypeSeq. What we know is that eugenia is in Myrti, whereas Syzygium is in Syzygiae, and it's completely different lineages. All right, here is an earlier work done before I started my PhD. And this is the phylogeny generated for Syzygium. And in the past, of course, there were a lot of air-like genera. When you have certain things with a fused calyx, it was called cleistocalyx, and etc., etc. So there were many genera then. But then this work shows that all these air-like genera are actually Syzygium. They are well supported in as Syzygium, and in fact they should be recognized as a different rank, perhaps a subgenus, and that's why in this Craven and Bafen paper, they recognize 6 subgenera and 7 sections. Nevertheless, these are all chloroplast genes. Much earlier works, Sanger sequencing, you can tell that Syzygium, subgenus Syzygium here, as pointed out by my cursor here, is that the relationship remains unresolved. And even the backbone of the relationships between other subgenera are unresolved. So therefore, I was given this opportunity to do this PhD, 4 years, fully funded, with funding for molecular work as well as my studies and school fees and all that, with 2 objectives. The first objective is to understand Syzygium subgenus Syzygium because that is the largest subgenus of the genus, and most of the diversity are actually embedded in Syzygium subgenus Syzygium. So what we know of all this richness from the Malesian region, I can tell you that most of these species are within this subgenus. So therefore, to better understand this subgenus, we need to break them apart further and try and understand a bit more. It's very difficult, but we are slowly getting there. And of course, the second part is the try and come up with a taxonomic enumeration, which I'm not sharing with this talk because the next bit, it's going to be very heavy, very complicated. So this is chapter 2 of my thesis, in which we talk, this is more a phylogenomic work of Syzygium that we have done. In this study, we included 292 samples, of course, 3 outgroups. So therefore, we have about 189 Syzygium tips for this phylogeny. And as you can see, we have about, if I'm not wrong, 68 authors, including myself, multinational. I wouldn't be able to do this work all by myself. It was a lot of team effort. It was a lot of collaboration, and it's not easy, but we made it, and we just published it last year. So one very important thing why we highlight the Syzygium grande, which I think you have it in Java, and is possibly also cultivated as a street trees, is that we generated a reference genome of grande, and we use it as an anchor in which the rest of the taxa mapped onto the reads. So here is a array of Syzygium that I have sampled from Australia to in Borneo, and Syzygium adelphicum is from New Guinea. That's when I went on a trip to Afak Mountain, a beautiful place. And then of course on the left, the grande images are actually the individual that I have sequenced for the de novo assembly. So what you are seeing here is the figure on the right is actually an assembly of the grande genome using mini ASM. Okay, these little cotton knot-like figures are actually individual assemblies. They are not terribly wrong or difficult, but we know for sure that it is much easier than some other groups. So in the same lab, we have groups that worked on mint. It is terrible because everything is in this figure, when they generated it, everything is not in individual pieces, but they are cuddled up like a fur ball, like a cat fur ball, you know, when you see it. It's just so difficult to untangle. So we were very fortunate that we have a very easy assembly to do. So this is a different assembly. And on the left is a different assembly in which we actually get the material and send it to a company in the States to do a high C analysis. And these two analyses, the de novo assembly that we use for mini ASM and the high C assemblies are complementary. That's when we know that our de novo is accurate, and we polish it and we work on it together side by side. So if you look at these figures, colored figures here, there are actually 11 little windows that represents 11 chromosomes of Syzygium grande that we obtain, and this is the haploid genome size of Syzygium grande. And then here, with all this deep information they are getting, so we have this full reference genome of Grande. We would like to learn a bit more about its ancient history, like looking into, whether there was a particular event in the past, like polyploidy. We can use with this is that here we are looking at synthetic dot plots. The top figure is where you have syzygium mapped with syzygium. So it is itself mapped with itself, the chromosome, the 11 chromosomes, and you get a kind of like a scatter diagram. Then at the bottom is where we take a published de novo assembly of another member in Myrtaceae, which is Eucalyptus grandis from Australia. We map syzygium and eucalyptus together, and we get this 1:1 portion, which means they are sharing similarity in a ploidy event. And so we kind of assembled members of Myrtales, which is Eucalyptus syzygium, Eucalyptus, syzygium, and punica. And if you look at the arrow, they are actually, and then when we lay the plots together, we can tell that they have a single polyploidy event. Whereas, and then of course, we must have a control. So in this work, we included two control of an other earlier work by others published. One is a vitis, which is grape, and the other being populus. Populus is known to have two polyploidy events, whereas vitis is known to have one. So if you look at the pink chart, the pink graph here, that's the single polyploidy event that vitis has gone through. And then if you look at populus, it has got two humps here of the two polyploidy events that populus has gone through. So if you look at this, we know that the order Myrtales has gone through a similar single event, different from the control. All right. So therefore, when we mapped syzygium, eucalyptus, and punica against vitis, we get an identical chart in the synthetic dot plots. So in the same study, we have proven in our work, we have proven that in an earlier work of what they call the 1,000 plant transcriptomes published in Nature, it was postulated that within Myrtales there were many individual polyploidy events estimated. But in our work, we have clearly shown that it is only one pan-Myrtales events that affects the entire order. All right. So with all that kind of work that we have obtained, we proceed with the Illumina sequencing of 30 GB. That's when you can imagine that first you have this very long train, a full train of Syzygium grande. That is our reference. But of course, if you want to do 289 or rather 292 samples and gone through everything the same, it will cost us a lot of money. So therefore, for this resequencing, for this 292, what we did was that we do a shotgun. So you just imagine that you have a train. Instead of a full train, we are doing some shotguns looking at different compartments. Okay? And then when you again imagine that you have all these 292 trains in a row, of course you have your reference at the top. Then you have all these, the rest with different segments. You will then try to align them according to the same segments that you obtain. All right, and the missing data from this portion would be what we would imagine. All right, so therefore we know we will have some missing data, but we know that we were going to align this train with some missing data. That's how we are doing it with this 30 GB of data. And then we call Mazuka assemblies, we call SNPs, and of course we have a chloroplast genome, but we know that the chloroplast story is a very different story and it doesn't tell a complete story, which I will show you. So here is a Mazuka assembly of all the 292 reads. We have very good assembly for most of the samples, except for those in blue and a bit of red. Okay, but as long as it's yellow, they are very good. Okay, so this is what I meant. The chloroplast would have a different story compared to the Basco. So the Mazuka assemblies will assemble all the Basco genes, and we use these genes, which is about 1,227, and assemble them and generate trees. So you have gene number 1 will give us a different model of trees. So all these 1,000 over trees we coalesced and generate one species tree. And what you are seeing here is a tanglegram comparing the gene trees, which is the species trees derived from genes of these 1,227 genes, against chloroplast, which is very, very conserved, we know. And if you look at the incongruence of the relationship, it's for sure it will tell us a different story. Okay, and then with this SNP tree, what I did is we have this time tree, and we laid it out. We have a fossil, and we mapped it. We used the fossil to serve as an anchor, and then when we kind of calibrate it, we discovered that almost half of this taxa in our phylogeny is very, very young. So meaning they actually appear at a much later stage and very rapid, which you can see from our tree later. So again, back to this Sunda and Shahu thing, shelf phenomena, these two very interesting land masses. Most plant species has this very conventional migration route that comes from west to east, all right? But for Myrtaceae and a few other families that I can't recall now offhand, they actually have this unconventional route where they actually travel from east to west. And this is clearly shown in our work later of syzygium migration route. So in BioGeoBase, we can tell, we have all our regions labeled. We can actually tell that they are traveling from Sahul at the different lineages all the way to Africa. If you look at the top plate here, okay, it originates from Sahul, it goes into Sunda, which is the turquoise color, and eventually it goes into Asia, mainland Asia, and into India and Africa. So here's the color. So we know the African syzygium, it's wrong for me to say, but I think in a layman term, it would be much younger in a sense compared to syzygium that occurs from Australia, for example, in speciation kind of age term. All right, so here is our phylogeny in a circular form. What we have now, it's a very, very well-resolved syzygium, subgenus syzygium. It remains the same, circlostratum and perikion. Acmina is expanded. Some of the earlier subgenus recognized by Craven and Biffen, we are going to move it, sunk it into a section in subgenus Acmina. Our recent paper, co-authored by Sebastian Hart from Kew, we have named a new subgenus called Oborapi from my molecular work here. So if you look at the tree, the black dots actually indicate that the migration or the invasion doesn't occur just once. but in fact, from our work, the invasion actually occurs 12 times from Sahul to Sunda. It doesn't just go on one wave, but there were multiple waves coming in from different lineages, from Sahul to Sunda, from Sunda onwards to India and Africa. All right, and then here we were just focusing on Syzygium, subgenus Syzygium, and this is the clade where I will go back to the previous slide, that if you look at the very, very short backbone here, it actually reflects a very rapid speciation event. And we try and amplify this particular portion into the previous analysis, where we use a PCA analysis and we look at this subgenus Syzygium of a particular clade, and we run it on a neighbor net, and we discovered that at the base of the clade, in fact, it is actually not a clear split, but you get this network that represents some introgression, incomplete lineage sorting that happened in the past. So this is our speculation. It is either that or hybridization of species that happened. All right, but we know that there's no hybridization occurred in Syzygium because we did the next test. So this is a PSMC analysis where we confirm that the biogeographical pattern conforms to what we have discovered in our previous slides, where it shows that the migration is happened very fast and very young. And then this is the F3 statistics where we did to trace hybridization. And from what we know is that there is no evidence of hybridization in Syzygium, and therefore the neighbor net that we are seeing actually are what we can say incomplete lineage sorting. So therefore the summary of this PhD work, of this phylogenomic work, is that biodiversity genomic can actually successfully be done. Of course, I must always say that you must have the kind of funding to enable you to do so. But if you don't have the kind of funding, then you can scale back and do a different phylogenomic study, not the one that we did, but the one that we did now seems to be doable, where you generate a reference genome, you generate short read draft and genome assemblies, and then you map it to the reference genome to kind of enable us to study this phylogenomic work framework. So with Victor, we are looking into other organisms as well. So we are looking at Fagrea at the moment, which are wrapping up, and we hope to publish the work. One of his students is looking at, and I help with a lot of sampling, most of the sampling, in fact. So we are getting some very interesting results there, and based on the phylogenomic work of Fagrea, that we'll be naming a new genus, in fact, from New Guinea, of Fagrea, which is from Gentianaceae. So therefore, I would like to kind of exceeded 5 minutes of my talk. Of acknowledgements, I have this principal funding from NTU and National Parks Board. My work has been generously supported by UM in the past, NParks from past to present, and NTU, Kew, Aberdeen, and Buffalo when I was doing my PhD. So thank you so much for your time. My sincere apologies that I've exceeded the time frame. I hope I can share with you briefly of what I have done and motivate you to pursue further in your work. Thank you.
Thank you very much, Dr. Low, for your presentation, and I hope this is in accordance to our running course now. We have the course of biodiversity and systematics of the phanerogams, and your work really mirroring our course. You have done many things in morphology, you've done many things in molecular, and you combine them into one large analysis. That's really in accordance to our course. So without any delay, I will share a question and answer session. For the student who wants to ask question directly, you can open your microphone. Otherwise, you can also address it indirectly from your chat box over there. I've seen 5 teaching staffs, Ibu Ratna, Ibu Pinta, Ibu Yani, dan Pak Rizky in our Zoom. However, I will ask student first before we move to a staff to enter the questions. Anyone have question already? Can raise the hand. Or you can write it down in Bahasa Indonesia or in English. I think Yewen can understand a little bit of Bahasa Indonesia.
Bisa Bapak.
While waiting for the students' questions, perhaps I will address a question from our program coordinator, Ibu Reni, who has left us because she has another appointment. She asked about the use of the use of branching in the classification of gardenia. Is it absolutely necessary, or well, in outside the Malaysian region you can have another character? And how about if we miss the character because of herbivores or because of human intervention, which makes the branching is not as natural as it was? That was her question.
Yep, I agree that that's why I did say that if you can see from my slides of Syzygium Gardenia jasminoides, I actually studied it from a cultivated plant, and you really need careful observation. So especially, you need to get the plant from the nursery and then leave it there and try not to do any pruning or any cuttings to it. It would be the same as the trees that we obtained. So I was very fortunate because I was part of the botanic garden in the University of Malaya then. And the gardenia that we purchased from the nursery, big trees in large polybags, we kept it as it is. And when we planted it, we actually didn't do any pruning at all. So there's no disturbance. And of course, in the university setup, then we don't have much destruction from animals, apart from maybe insects and whatnot. But otherwise, we don't have any herbivores. So I think in that way, I was very fortunate, and I was able to observe this particular branch of study, which is the growth architecture. I don't think anybody has been doing it apart from in the lab where I was from, because the professor was very keen in promoting this very branch of study, which is the tree growth architecture. As I said, when you fully understand this at a very young age, like when you have understand it is in the lab, like how it grows and all that, when you go to the field, you would be able to catch it and grasp it that, yeah, even though there will be some alteration by maybe animal, or like in the forest, it could be due to a tree fall that breaks a branch, or it could be due to a climber that, affects its growth and canopy, how close it is. But you will still be able to catch the very basic units of the growth assembly of the particular species. But as I said, it won't be a very good taxonomic tools to be used to distinguish species apart, but it would be very good if you can use it for a particular group. So in a sense, I can use it for gardenia in the sense that they will have shrub with a different kind of growth, and the species that form trees would have a different kind of growth. That's about it. There's no taxonomic whatsoever values to it, but it's good to learn a bit more on that.
Thank you very much for your answer. Anybody has question for Yuan? Wilson? You question a lot, Wilson, normally.
Maybe perhaps I mean, I was a student before, so this will be a moment of time where I will always keep quiet, because especially when the lecturer says, do you have any question? We always say, oh, I keep quiet, I mean, it's normal, but I really do hope that you can, I mean, students can see what kind of work that we are moving into. I hope I can really motivate you. And the most important thing I have always shared with a lot of students is that nobody can help you if you do not help yourself. So the motivation is very, very strong if it comes from yourself. So like I said, I have always been very keen in plants, and I have been pursuing it. I'm sure most of you are very keen in plants, but it's just maybe a different branch or different kind of science that you are interested in. But I'm happy to answer any questions if you have any. It's just very keen to share with most of you.
There's one question in the chat room, I think.
Of the many plants, what species are very wide distribution in some countries with similar characteristics? I think it's it's very different approach if you look at it. Some plants would have very wide distribution. I would say gardenia, if you look at the genus itself, it is very wide. But if you look at the species itself, they can be very, very narrow. Why? I'm not sure. Maybe it's later speciation event, and then now with all the geographical barriers, it doesn't go out from there, or it could just be due to dispersal. I think, but some plants are really rare.
Bu Ratna, you have more comments about the answer? I think it's quite clear. We have another question from Nur Wahyu about Ibu Ratna. I have a question for you, sir. In researching the Gardenia genus, were there any special differences between each species? That's right above the last question. You're still mute.
Sorry, can you hear me?
Okay. Yes.
Yeah. No, I think the question is yes. Different species of Gardenia would have specific differences, just as what I showed of the calyx morphology. So that is one part where you can use to distinguish them. Of course, we can't use the tree trunk character because they all have smooth and they have smooth bark and no buttresses, so that's very common. But we can use the calyx feature for that to distinguish a species. But of course, different groups of plants will have different groups of different characters to distinguish. For example, if you look at orchids, flower will be very important. Then the different kinds of flowers, and then maybe begonia would be different as well. So different groups of plants would have a different characters, I must say. So calyx doesn't just you can't use calyx for everything. So it may be good for gardenia, but it doesn't work for syzygium. So you can't use calyx to distinguish syzygium apart because they are all very similar.
Do you have further question, Wahyu? No, I think. So other question. Are there other questions? Oh, enough. Okay.
There's another question I saw is from Muhammad Ridwan Hafiz, and it says, what could have caused the rapid diversification of syzygium? It is a very good question. To be very honest, we are still not sure. We do not know what is the cause of this very rapid speciation, but we know that there is this event happened. But what is causing it, we don't know yet. It's something that we are very keen to look into and do a bit more work to it. That's for sure. If you remember that I have a very good, very resolved phylogeny of syzygium. Now what we are working very hard on is to try to recognize or to find the characters that group these lineages together. Yes, now we have a good tree, a phylogeny, but you now need to find characters to distinguish them apart. So, and I can tell you, it is very difficult for syzygium to look for characters.
Some more comments from Ridwan Hafiz for the answer from Dr. Low? Because I also have question. Wait. Thank you, Dr. Low. following Ridwan's question, because I saw you that your cladogram show that the Myrtaceae, the whole Myrtaceae, somehow derived from the Gondwana,
Mm-hmm.
right? And the only genus which derive in Sahul is the Syzygium. Is that correct or not?
Well, in the phylogeny, we were only looking at Syzygium. If you are referring to the work that I did, we were only looking at Syzygium, with a bit of other genera as the outgroups. But if you look at the work by Eve and other works by Thornhill, it has been proven that the entire family of Myrtaceae is derived from Sahul, from Gondwana, and actually goes from east to west. Has a very unique distribution compared to most plants where like Dipterocarp we know is coming from west to east, where there's a rift from India that hits Himalaya. Oh no, from and then from there it species travels down into the Malay archipelago. But for Syzygium is very different. For Myrtaceae, in fact, is very different. That's why you see the diversity of eucalyptus. It's very rich in Australia, and of course other Myrtaceae genera there, Melaleuca and whatnot. Whereas if you look at this part of the world, we have only very few species. But Syzygium, in a sense, is very successful. Why is it successful? It could be the dispersal methods, because Syzygium has got fleshy fruits dispersed by birds, by bats.
Right.
Syzygium grande, I'm not sure whether any of you have observed. I'm sure you would have seen it. Syzygium grande, the fruit is maturing green, and is actually dispersed by bats. So here in the Botanic Garden, I will always find Syzygium grande fruits on the floor, because at night the bats will carry the fruits from somewhere, and then it will rest on our roof just outside the herbarium. They will eat the fruits and they will drop the seed on the ground. So I always get this and the fruit that they collect, the fruit of Syzygium grande collected by the bats, are actually green. So I think we know the success is through dispersal by animals, birds, and mammals, for sure.
Right. That's amazing. I mean, the only genus involve animals is only Syzygium. That's never crossed my
Yeah,
mind.
in South America is Eugenia. So the
Right.
same pattern is happening in Eugenia. Of course, we still have Eugenia, but we only have one species in Sundaland, which is Eugenia rhinewatiana, which is occurs around the seashore. There could be a few more species in New Guinea, but we won't have more than what is happening in South America, where Eugenia is extremely rich. Eugenia is another genus with fleshy fruited. So if you look at all members of Myrtaceae, there are only two with fleshy fruited species, which would be, I mean, the big ones, Eugenia and Syzygium. In the past, it is speculated that they are closely related, but then now we know they derive independently the fleshy fruited bit of it, and they're even from two complete different lineages. Eugenia is from Myrtae, Syzygium is from Syzygiae.
Very interesting. Yeah, especially that connection between Syzygium and animals. That's amazing. We need to collaborate more with people with understanding in ecology or
Yep.
interaction between animal and plants, I think.
Yep.
Any other question from students or teaching staff? Unita, that's a question from Unita, Dr. Low.
Yeah. What was the best experience you had while studying gardenias, and what species do you think personally really interests you, and why will they really interest you? So, to be very honest, uh, uh, gardenia. When I was studying gardenia, I was really puzzled by how the fruits mature. If I may show you, let's go back to the slides. So let me just go back to gardenia. All right. So what really interests me is Jasminoides. Uh, it is not from our region, but it is very interesting. Why? So if you look at, can you see my cursor? So, so here I have my cursor on the fruits of Gardenia jasminoides. And then just below it, I have a fruit of Gardenia tubifera, which you also have it in Indonesia. What you are seeing here is a display of the seeds. Here you have a mature fruit that splits open uh, uh, and displaying the seeds embedded in a red pulp. This is a mechanism for dispersal where the seeds are flattened for gardenia and they are actually eaten by birds. And then the birds will fly far away and then they will poop and then the seeds will grow. So the seeds will survive through the gut of the birds. But for Gardenia jasminoides, it was only very late of my study after I've written up my thesis that because I've never seen it, it's not native to our country, it's not native to Sundaland. I realized that the fruits actually does not split open like our native gardenias, like what we are seeing in Gardenia tubifera. I have seen all our native gardenias, they all split open to display this delicious fruit for the birds. But for jasminoides, apparently it doesn't split open. And most interestingly is that if you look at tubifera, that my cursor now is showing you, this is the part which we call the stone, which means it is very, very hard. And this part is actually missing in the fruit of Gardenia jasminoides. Why? I have no idea. And it was only very much later I realized that when Gardenia jasminoides matures, when the fruit matures, it stays that way and it becomes soft and squishy like a soft cake. And I believe it would have a different kind of mechanism, dispersal mechanism, different thing. Something would be eating it in the wild where it's native, but I just don't know. Something. I know for sure our gardenias are all eaten by birds, but not jasminoides. So I'm still very intrigued with jasminoides, just maybe because I've never seen it in the wild, and I would really want to go to. I've never been to southern China or southern Japan to see it in the wild. It occurs as a bush in a forest, in a seasonal tropical forest. So yes, Gardenia jasminoides is my answer. Thank you.
Any comment from Unita? Oh, thank you, Dr. Lo. So, yeah, we have a very interesting presentation and question and answer, even though students are quite shy to address their question directly, but questions addressed in written, I think it's as beautiful as the direct question. So I have to thank students that are willing to address their thought and raising their hand to ask question to Dr. Low. And I appreciate that Dr. Low were willing to answer the questions. I guess now I have tons of questions also in my head, but it's not ethical if I ask my own question and we have a personal chat here in this forum. So so I think Dr. Low will be delighted to answer a personal question directed to him personally. You can see his email in National Parks of Singapore website. I did that also because I used that email which I copied from the website, and I was very glad that Dr. Low answered my email, even that we've never been acquainted before. So I really appreciate that. Due to our time restriction, I think we called the question and answer a day. Final question from Unita has concluded our question and answer session. So I again appreciate and thank Dr. Low for being with us. I have to thank students for listening and sharing your interest to our plants in general, and our native plants in particular. I also have a wish to thank teaching staff that I recorded at least six staff attending this talk. So it was amazing well experience. So sharing Dr. Low's experience also enlarged my experience, and I hope this will also enhance the students' will to explore more of our own biodiversity. We have Java, we have Sumatra. Dr. Low had been to many of our islands. Why don't we use his experience for our own experience? I think that's a good idea to all of us. So allow me to conclude these two sessions, the presentation and question and answer, and I have to return this Zoom to our announcer, Tatu Mutiara. This is yours. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much.
Yes. That was such an amazing material, and also Q&A session that we had earlier. But sad to announce, we are now in the end of today's lecture. I would say thank you very much to Dr. Yuan Lo for an outstanding material you've shared with us today. It is so amazing, insightful, and also inspiring out. Thank you very much, Mr.
Thank you. Thank you so much.
Before we close today's event, please, all audience, open your camera because we will take the documentation first. Yes, operator, are you ready to take the pics? So please do the count.
Okay, I will pick the picture. It's been 2 slides on the first page. 1, 2, 3. Wait. The second. Okay, 1, 2, 3. Okay, done.
Okay.
Thank you so much.
Thank you. Okay, thank you all lecturers and also all the audience. To close this, let's say Alhamdulillah. Alhamdulillah. And thank you so much for your full attention to this amazing event. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Allow me to leave this Zoom. Dr. Low, I will contact you later by email and WhatsApp.
Thank you so much, Agung. Thank you so much, everyone. Bye-bye.